All right. Hi guys, we're gonna get started. This is topic six. Oh my gosh, topic six, not sick. Microtubules, structure and function. But we're gonna go over basically just the first half of this week. So everything up until like drugs, naps, motors, things like that. That'll be covered on Thursday. So if you're more interested in that, you can wait till Thursday.
Okay, so we have three types of cytoskeletons in eukaryotic cells. The first one is actin filaments, and like it's a subunit of microfilaments. And this one is mostly for movement in the cell. Then we have microtubules, and we have the intermediate filaments, which are more for just mechanical strengths in the cell.
Okay, so going about each one a little bit more specifically, the actin filaments, or I guess in general in this class, you don't worry too much about much of them other than like the microtubules for specifics, but it's good to know about these just in case she asks. So actin filaments, also known as microfilaments, they do a lot, but essentially they have to do with movement, and specifically like the cell movement. So actually being able to move the cell rather than like moving inside the cell, which is more microtubules. The actin filaments are polymers made up of monomers. So that means like there's a lot of small individual pieces that make up as a whole. So these like globular actins make up, come together and then make up like the full filament. And then they can serve as a track as well for a family of motor proteins called myosins. We'll talk about myosins not today, but then later in the week on Thursday, that's going to be covered there.
Okay, in microtubules, as Tina said, they're more for like they have to do with movement in the cell too, but like it, as Tina said, actin has to move like to do with movement of the cell itself, but microtubule is movement in the cell. So that's where we have dynein and kinesin, which if I memorize them like kind of like the cat walk. They do it in the cell. And they function like in the cell division by forming the spindles, right? Yeah, they form the spindles where they ball the chromosomes. And they also in a neuron, they act as like a track. So they move the vesicles from the cell body, which I guess you don't need to know it for that, but they like serve as a track in the neuron. And they're also polymers, and they're made of monomers, which are called tubulin, which I guess here it would be like, that's a tubulin dimer. Tubulin are made of like alpha tubulin and beta tubulin that makes like one tubulin, which makes like when they all come together, it makes a microtubule.
Okay, and then the last type of like cytoskeleton-esque part of a cell would be intermediate filaments. They don't really serve for movement, unlike microtubules and actins, but instead they're more structural. So they keep like the cell in the shape that the cell should be, making sure everything's sturdy. Especially popular, I believe, in like muscles as well. So they help bear a lot of tension and maintain the cell shape and other organelles in the nucleus.
All right, so now we're gonna talk about how could we visualize the cytoskeleton in microtubules. What we could use for that is fluorescent microscopy. And the way it happens is that we tag tubulin with green fluorescent protein, and then we put it under the microscope and we can see like a glowing green structure, which shows the tubulin on the microtubule.
This is like, I think one of the questions on the past exam was like, oh, like, how would you visualize something? So
that's the answer.
Hopefully you guys said something along the lines of like fluorescence or GFP or anything like that, just because it's really hard for you to obviously see anything with like your naked eye. Even through a microscope, if you look through a microscope, you just see like black and white or essentially. But if you use fluorescence like GFP or anything that glows like that, other examples would be like DAPI or there's other things, or like potion or things like that. But yeah, by actually dyeing it, then like each of these parts will glow, and then you can control like if you want to see DNA, if you want to see microtubules, if you want to see like the nucleus, anything like that. Yeah, exactly. One of the classes at this school that's offered is like bioimaging 499 or something like that. Really cool class. It's a fluorescence microscopy class. And this is like essentially what we do in the class. You learn how to like microscope and actually use a microscope and like see different parts of the cell. Very cool. It's a hard class, but very cool. But yeah, this just shows some more examples of fluorescence microscopy. So in the green, you see a little more like microtubules. The yellow is the Golgi body, the blue is the endoplasmic reticulum. And it's just cool because like it's one thing to see like a cartoon version from like a biology textbook of like, oh, this is the ER or whatever. And it's another thing to see like, yeah, to see how it is in real life.
Yep. All right. We're gonna like dive more on the subgroups of microtubules. So there are 3 subgroups: the ester, which grows from the centrosome and attaches to the cell cortex. And then we have the kinetochore, I don't know if I'm saying it right, kinetochore, which also grows from the centrosome and attached to the kinetochore. And then we have the interpolar, which grows from the centrosome and it overlaps the spindles in the cell cycle. And this is used during the spindle assembly and chromosome separation during M phase and cell division.
All right, so talking just a little bit more about microtubules, specifically their structure, so that you guys fully understand like what microtubules are made up of and like how they function. As Miriam had talked about a little bit before, they are made of tubulin heterodimers. So that means there are 2 parts to one dimer. You have the alpha and then the beta subunit. Where is it in this picture?
Alpha's a blue and beta's.
Oh, yes.
Yeah.
So alpha and then beta. So then like 2 of them come together and then that makes like one singular dimer. And then a bunch of these tubulins make up an entire microtubule. Microtubules are polar molecules. So that means they have a positive and a minus end. So in the picture, you have the positive end or the plus end as it's written over here, and then the minus end down here. And it's different in the sense of like, it's not polar charged or anything. So like, don't think about amino acids in this case. It's literally just means that one side has more tubular addition or like a lot more activity happening, and that's the plus end, whereas the minus end, not as much activity is happening. And then we'll talk about like why that is in a little bit, but also, tubulin is a GTPase, so that means that the alpha and beta subunits are bound to GTP. However, only the beta subunit can be hydrolyzed. So when it's bound to GTP, there's a lot of good affinity and you'll get addition. But when it hydrolyzes from GTP to GDP, that's like bad affinity for each other and they all disassociate. That's like what the whole catastrophe thing that Laura talks about is what about. Yeah, so then all the tubulin come together, they form 13 protofilaments. So each protofilament is like this one singular kind of like line in this picture. So that's like one protofilament, and then 13 of those make one microtubule, and then it's hollow on the inside. The inside is called the lumen. And then again, the minus and the plus end. So usually, and if you're in a live cell, the microtubules, the minus end is put into the microtubulizing or microtubule organizing center, or the MTOC. That's the centrosome. And then on the centrosome, so when you have like cell division, you get like the 2 centrosomes on either side of the cell. And then the minus end is plugged into those centrosomes. And then the plus end is like the spindles that you see coming out of the centrosome that you would see during like cell division.
All right, we're gonna now discuss dynamic instability. And so that's basically talking, as like Tina said, about like GTP hydrolysis. And so again, like Tina said, we have the plus end and the minus end in this case here, we're gonna focus more on the plus end. That's where we have the most activity. And as we said that it's GT based, and that's this is the only end on the microtubule that hydrolyzes GTP. And so we could have two scenarios. If we have GTP hydrolysis happening at a greater rate than tubulin addition, then we're gonna get depolymerization, and we're not gonna get a GTP gas. So we're just gonna get the tubulin is gonna break down. But then the other scenario, if we have GTP hydrolysis is happening at a rate slower than tubulin addition, then this is when we get the GTP cap formed, and we're gonna have the microtubule like more stable in forming. We're gonna like have elongation of the microtubule. So the main idea here is microtubules can grow and shrink independently of each other. Do you have anything?
Also, if you have any questions, feel
Yeah.
free to just like shout it out. Or if anything that we say don't make sense. Okay, so I touched a little bit upon the MTOC briefly, but this is to explain it a little bit more because they can be very confusing, and students often get confused about like what the MTOC actually is or like entails. So MTOC stands for microtubule organizing center. They're needed in the cell because a microtubule, it's really hard for it to just kind of like spring out of nowhere and grow de novo or like from nothing. And so the MTOC allows kind of like a starting point for the microtubule to grow from. So in this example, you have the gamma, or sorry, it's the gamma, I don't know what gamma tubulin small complex assembly. So it's like these two together, and then a lot of the small complexes together ends up forming a ring, which is seen over here. And then once this ring forms, it's like nucleation, and then you can start to have tubulin get added easily, and then the microtubule itself grows. So in a cell, for our purposes of knowledge, this is the centrosome. Inside the centrosome is the centrioles. On top of like kind of the surface of the centrosome, you see in this picture, it's like red rings. Those rings are your gamma tubulin ring structures. And so from those like nucleation sites, that's where the minus end is embedded within the centrosome. And then all the stuff that's sticking out, those are the plus ends of the microtubule. So hopefully that like clarifies structure a little bit more.
Can I ask for some clarification?
Yeah.
Questions? So the microtubule grow only on the plus side?
They can theoretically grow on both sides, but because the minus end is like in the centrosome, there's not much happening there. So for like our knowledge, like we just focus on the plus end. Yeah.
Okay. And the gamma tubulin, is that only like involved in the MTOC?
Yes.
Okay.
Because regular tubulin, the alpha and beta tubulin is the microtubule itself, but the gamma tubulin is for like the nucleation in the ring site. Yeah. Yeah. For the question you said about like the minus and plus end, that's more relevant of like, oh, both sides could be added or subtracted when you're in like in vitro, like in a test tube, which we'll talk about in a bit as well.
Okay.
All right, so yeah, now we're gonna talk about in vitro. So in vitro as like outside of the cell. So when we're working on a lab, we can just work on a tube, and this good. I don't know if it's, wait. Okay, that's more slide.
Oh yeah.
So we can add there your tubulin and GTP, and this will give us more of like polymerization, or we could also add like more of like tubulins that is bound to GTP, and this is gonna give us depolymerization. So like the good thing about it, about like doing experiments in vitro, is that you have control over what you're doing. So you could control the concentration of how much you're adding of what protein or what molecule. However, the negative sides is that what happens in a lab or outside of the cell does not necessarily mean that it happens inside the cell. So when we work in vitro, we just have to make sure that what we're doing is aligning with what would happen in a cell. And that way we can apply in the cell.
Yeah, exactly. Because like when you're in a test tube, literally all you put in is like tubulin, GTP, and then like maybe a drug or whatever that you want to see what happens, but you don't have like the rest of the organelles that a cell normally has. So you'll never know like 100%. Oh, like maybe in lab or like in an experiment, like this drug works amazing. And then you actually put it in a person. It's like, why didn't it do anything? And it's because like you can't control for everything in a body, like inside a test tube. Yeah.
Because we always have like other enzymes or inhibitors, molecules that might inhibit that drug.
Yay, biology. Okay. Okay, going into microtubule dynamics in vitro, because this experiment is talked about a lot. And so you definitely got to understand like how an in vitro assay. Okay, I never can pronounce that word. How an in vitro assay works, because more likely than not, you'll definitely see these either, if not on just the problem set, then you'll definitely see it on like either the next exam or the final exam. So good to understand. Holistically, just to go over it generally first before we get into like the different parts of it. The microtubule dynamics in vitro is a time course experiment. So that means it happens over a period of time. And we use the way that it works is that it uses a spectrophotometer. Yeah, and basically what that does is like it calculates absorbance level over time. So like the more tubulin or like stuff in the tube you have, the more absorption you're going to have, or like the more light that's going to be absorbed or like calculated for. So that's why when you have like elongation or like you have over a period of time, you're going to have a higher absorption because there's more stuff in the cell now. Whereas in the beginning, like not a lot of microtubules form, so you're not going to have absorption. And that's just like the method of how they do it. But yeah, so you get absorption versus time. The more microtubule present, the higher the absorbance reading. And then there's 3 phases typically seen: lag phase, elongation, and plateau.
Okay, now we're gonna talk more in depth on the lag phase. And so as Tina said, that it takes time for us to form the microtubule. So it happens over like a period of time. It doesn't just happen like right away. And since we talked and said that microtubules happen like from scratch, from nothing, we start with the lag phase, and just because we don't have a lot of tubule or microtubules formed yet. And so the order of like the formation of microtubules is, first we have the pure tubulin, and then we have the dimers form, which is the alpha and the beta dimer of the tubulin. And then we have the oligomers. I don't know if I said that. And then we have the protofilaments, which I don't know if I can find in the slide before.
Yeah, it was like that picture of
yeah, you
all the
showed
way
it.
near
It
the
was,
front. Yeah.
oh, right here. Yeah, this right here is the protofilament. And then we have a lot of like these 13 of the protofilaments coming together to form the sheets. And then that sheet is like gonna close and form the whole tube, which becomes the microtubule. And then once that happens, that's where that like we have the elongation phase. And so the higher the free tubulin we have, then the shorter is the lag phase. So we're gonna have elongation happen faster. And I have a question. What do you think would happen if the seeds already existed? So we don't have to still make microtubules from scratch if we already have some seeds there.
Probably wouldn't be a lag phase.
Yes, exactly. We're gonna probably just start at the elongation phase.
All right, so into elongation phase. So this is when the formation of seeds leads to the rapid addition of tubulin binding. So like once your seed forms, all the GTP is there, like the cell or the tube is in perfect condition to just add it on and get binding. There's a really high affinity for each other. And so it's just going to shoot up and like into this kind of exponential is the word, kind of like this exponential curve all the way up. And it happens primarily on the plus ends. And I guess I kind of touched upon this a little bit before, but do you know like why it would mostly be on the plus ends rather than like plus and minus ends?
Well, because the minus end is embedded the NTU state, and also the plus N has like the exposed beta.
Yes, so that's part of it. If you were in a real cell, then the minus N being inside embedded would be valid. But technically, like because it's in a free tube, or like in a test tube, there's no like centrosome or anything like that it's embedded into. So theoretically, you can have what's it called? Polymerization and depolymerization on both ends, but it's just that the plus N has a greater affinity for GTP. And it's like you said, it has the beta, that's the side that has like the beta tubulin, which is why like it has a greater affinity. And when you have like a ring of GTPs, it's just going to stick together more, not only like stick together horizontally, but also like laterally. So it'll stick together as a ring and grow faster, like longitudinally as well. Yeah, and then just to kind of explain like graphically what's happening. So as your microtubules elongate, the concentration of free tubulin that you have is going to decrease, because remember in the beginning of the experiment, you only put in like an X amount of tubulin. You're not in a cell where you have like an infinite amount of tubulin. So eventually like you are going to start to run out of free tubulin. And so that affects the rate of assembly to decrease as well.
And this is why we get the plateau, because we only have like a finite amount of free tubulins that we could add that is not bound to GDP. And then at the plateau, then we're gonna have depolymerization and polymerization happening at the same rate. So it's not that there is no, like that it's not still or there is nothing happening. We're just having equal rates happening at the same time. And so we're gonna have hydrolyzation catching up to the tubulin addition. And we're gonna have more of the tubulin that is bound to GDP than GTP. And once tubulin binds to GDP, there is no way for us, like in vitro, for it to bind again and form like a microtubule on the microtubule plus end. And eventually we're gonna have all of them. We're, like we said, we're not gonna have any free tubulins. They're all gonna be bound to GDP.
And then like Mariam said too, it's like in a test tube, there's no way for us to change GDP to GTP. So eventually like they're all going to be GDP bound. But if we were in like a live cell, would this be the same thing, or would you have like a different end result?
Probably different, because the live cell probably has ways to get more GTP.
Yeah, exactly. There's just like I don't know the actual mechanism, but Laura's always just said that there's like a lot of GTP in the cell. There's just like an abundance of it and it's always around. So yes, nice.
I have a question.
Yeah, let me just wrap this up really quick. So just for like Zoom purposes, I guess if on Zoom you have any questions, feel free to, you can chat it or raise your hand and you can like speak out your question as well. But we're gonna close the recording for now. And then we can like answer any questions, or if you guys have like problem set questions too and stuff.